Okay, thank you for the introduction. So this is a first step of a work that is developed between the lab where I'm working, the Institut Curie, that is a microfluidic lab, and the diagnostic and pathological lab in the Saint Louis Hospital here in Paris. And this is about droplet microfluidics. In particular, droplet microfluidics represent one of the most promising technology for biological application. That's because thanks to the microfluidic channel, it's now quite easy to generate emulsion where the droplets are basically really monodisperse and where all these droplets are completely independent from all the other droplets in the system. So every droplet can be considered as a single reactor. And it's also easy to work with droplet at kilohertz rate. So it's easy to generate and manipulate this droplet at a really high rate. This is a workflow of a typical microfluidic digital PCR technology that is also now commercial available. Here in the first line, you can see how the droplet can be generated, and then usually after the droplet are collected in a tube, a normal PCR tube for the amplification, and then finally they are collected back inside of a microfluidic chip for the detection. This kind of approach is really interesting from statistical point of view, but can be some limitation regarding, for example, possible information about the droplet that are losing. For example, in this way, we can see how the droplets are completely mixed in the chip, but even in the tube. And so we lose some interesting information in the droplet if we put different things inside of different droplets, for example. And then we have only one phase, only the liquid phase. There is not possibility to introduce a solid support in the droplet. That's why we want to move on. Oh, yes. In a different direction. So we want to work with a droplet that are in confined state. So it means that the diameter of the droplet is smaller than the size of a channel. In this way, we have a deterministic order of the droplet. And we want to have the possibility to manipulate the solid state inside of a droplet, let's say magnetic beads. For doing that, we have to pay a price, and in this case, it's the throughput. We cannot work with our technology at really high throughput, at kilohertz. We are more in the hertz rate. But we think that this kind of approach can be really interesting for diagnostic application. And in particular, here I'm showing you our platform for the RT-PCR application. In particular, what we can do in droplet, it's at first start from total RNA sample, capturing the mRNA using magnetic beads, washing these magnetic beads, and transfer these beads inside of a solution containing the RT-PCR mix, and then perform the reverse transcription and the PCR amplification inside of a droplet. For at the end, go for normal qPCR outside of a droplet. So this is the platform that we are using. Is pretty simple. Basically, there is a syringe pump and that is synchronized with a robot for the generation of the train of droplets. And then these droplets are driven between two magnetic tweezers that we use to manipulate the magnetic beads inside the droplet. And then finally, the droplets are driven to a homemade thermocycle that it's adapted for droplet technology. So if we go step by step, this is the robot that we are using, and this is the scheme of the robot. I think. Yes. Okay, so basically we fill our microtreater plate with different solution, and we pipe it using a capillary, the different droplet spaced by fluorinated oil. And in this way, it's quite easy to generate a train of droplet where we control exactly what is inside of every droplet. And this is also easy to play with the volume, for example, of a droplet. Here, for this platform, we are using droplet of 200 nanoliter, but we can pipe it smaller or bigger droplet. So once that the train of droplet are generated, we move to the magnetic tweezer. The picture of the magnetic tweezer you can see here. Basically, it's just a a coil where we apply a current. A current generates a magnetic field at the center of the coil that is amplified by the two magnetic tips. And here you see the capillary in red where the droplets are flowing. And we can see the behavior, how it works. So when the droplet with inside the magnetic beads is coming in between the magnetic tweezer, we can switch on the current, and so switch on the magnetic bead, the magnetic field, and capture the magnetic beads. And when the second droplet is coming, we can switch it off the magnetic field, and so releasing. The movie is stopped. Yeah, okay. So when the second droplet is coming, we can switch off the magnetic field, and in this way we release the beads in the second droplet. So this is exactly what we can do in a tube, for example, but using droplet microfluidic. So using this platform, we perform RT-PCR, and for doing that, we generate a train of droplets where the first droplet contains magnetic beads coated with oligo-DT in order to capture the mRNA. The second droplet contains the sample, in this case a total RNA. Then we have 3 droplets of washing buffer, and then finally a droplet containing a mix for the RT and the PCR. So the first part of the experiment is just the capturing of the magnetic beads and transferring them to the second droplet containing the total RNA. Here, after 5 minutes of incubation, we are so capturing with an efficiency around 90% the mRNA inside of a droplet. After that, the beads are again captured by the magnetic tweezer and then transferred to the washing buffer in order to wash the beads, and then finally transferred to the final droplet where there is the RT-PCR mix in order to perform at first the RT and then the reverse transcription and then 15 cycles of PCR. These 50 cycles, they are there just to enrich the solution inside of a droplet of cDNA. At this point, inside of a droplet, so we have just cDNA that we can easily put our droplet in a single normal PCR tube and go for qPCR. These are the typical curves that we obtained from the qPCR machine. We tested with this platform at the moment 3 different genes. The first gene is the beta-actin, and so here you observe the fluorescence as function of the cycles for different concentration of total RNA inside of a droplet. In particular, we measure concentration between 2 nanogram and 0.2 picogram. We had always a signal, and we have a limit of detection around 2 picogram. You can see here the CQ value as function of the concentration inside the droplet that is linear basically until 2 picogram. Maybe it's interesting at this point to underline that the quantity of total RNA inside of a single cell is around 20 picogram. So then we tested the TBP, another gene that is used as a housekeeping gene in diagnostic lab. And even here we have a good sensitivity for 20 picogram. In particular, in this case, we didn't observe any signal for 2 femtogram, but we have we have good curves, and we are working on that. And then finally, we tested the gene that is interesting for breast cancer, the HER2. Again, we work between 2 nanogram and 0.2 picogram, and even in case we have a good curves until 20 picogram. And so we have a good results concerning the quantification. So finally, if we want to compare our technology with the standard technology that are used in the normal lab, we can say that we reduce 100 times the volume, because inside the droplet is around 200 nanoliter, and inside of the tube people are working between 10 and 50 microliter. Then we reduce the quantity of total RNA that we need to make an analysis, because we can work between 2 nanogram and 20 picogram. And what is really important for diagnostical things is that we reduce a lot the risk of contamination, because our system is a closed system. Once that the droplet is generated, the RNA inside of a droplet is not anymore in contact with the air. We are not working in controlled environment. We are working in normal chemistry lab. And this is really important so for the risk of contamination by RNAs. Then we have a good reproducibility because we have standard deviation less than 2%. As I show you, we are sensitive at single cell level, and from the technological point of view, we think that this magnetic tweezer can be really a versatile technology in order to introduce a solid state inside of droplets. That is something a bit new for droplet microfluidic, to manipulate especially magnetic beads. And then finally, as a future perspective, now are ongoing some clinical validation with breast cancer samples to detect ATR gene amplification. And from the technological point of view, we would like to work for integrate all the different parts that I show you in one single microfluidic chip in order to have something that can work maybe at more high flow rate at what we are working now. And then finally, we would like to integrate even the qPCR step in droplet. Finally, I want just to thank all my group in the Institut Curie, the collaborator in St. Louis Hospital, and finally the supporting of the work. There are different projects involved, and in particular the Foundation ARC that is supporting my fellowship. And you for the attention.
Thank you very much. Any quick one question before we go for a coffee break? If not, just grab the speaker when you go for coffee, and he is forced to answer all your questions. Thank you very much.